Hello, good morning everyone. This lecture is an aspect of ecological restoration and biodiversity conservation class. So students, download this video lecture and watch. I hope it is going to help you understand the notes that I have given you. The course code is LARR 402. Ecological Restoration and Biodiversity Conservation. I'm Austin Asari, a lecturer at University of Energy and Natural Resources, Department of Environmental Management. Let's look at the course outline. In this course, we will look at what is diversity, varieties of scales in which diversity can be considered. Why is diversity measured? We will also look at diversity indices. Here we will look at the different indices that can be used to calculate diversity. Now, whenever we want to assess reclamation success in a reclaimed man site, we usually measure vegetation attributes such as species composition, species similarities, and diversity to provide data that are relevant to management. Why do we do this? We do this because we always have management or conservation goals that pertains to a particular targeted plants community, and this can be described in terms of composition or diversity of species. What is diversity? Diversity refers to variations within or among living organisms on a site. Diversity is a variation within and among life forms on a site, or in an ecosystem, or on a landscape. Diversity could also be defined as the distribution and abundance of different plants and animals within an area. The word diversity is comprised of two components: richness and evenness. What is evenness? Evenness refers to how equally abundant species are in a particular area. So it means that the more equally species occur in proportion to each other, the greater the evenness of the site. A site with low evenness is an indication that one or more species dominate that site, while other species are relatively underrepresented. In an ecosystem, we don't want a situation where one or more species dominate the site. Before we continue, let us try and understand the differences that exist among these terminologies, because we'll be using them in this lecture. Abundance. What do you mean by abundance? Abundance is the total number of organisms in an area. It does not matter what the species, it does not matter what species they are, but the total number of organisms in an area. Relative abundance is how abundant a species is relative to the other species in the community. What then is species richness? Species richness is the number of different species in an area, or the number of species per unit area. Species richness does not matter how abundant they are, but the number of species per unit area. What is species diversity? Species diversity, when we are talking about species diversity, here it incorporates both the number of species in an area and the evenness of their abundance. That is their relative abundance. The next is scales of diversity. There are varieties of scales in which diversity can be considered. We can consider diversity in terms of genetic, taxonomic, in terms of life forms, in terms of functional groups, in terms of landscape. What do you mean by genetics? Genetics, we are talking about allele frequencies. Allele frequencies. In terms of taxonomic, we are referring to families, species, subspecies, and varieties. In terms of life forms, we are looking at it from the perspective of grasses, forbs, trees, mosses, shrubs. That is the categorization or classification of diversity in terms of life forms. We can also look at it in terms of functional groups. We are looking at it in terms of deep-rooted. We have deep-rooted species. We have nitrogen-fixing species, species that are able to fix nitrogen. We have species that are shallow-rooted, and we have those that are deep-rooted. We have evergreen species. We have soil crust species. Because these categories of species have different functional roles within the ecosystem. What is it about the landscape? Landscape, we are looking at the spatial patterns of vegetation at the landscape level. Why do we have to measure diversity? Is it important at all for us to measure diversity? Diversity measurement is very, very important, because diversity is often measured because high diversity is perceived as being synonymous with ecosystem health. In general, diverse communities are believed to have increased stability. Diverse communities is believed to be more resilient, to be more resistant to invasions, to environmental perturbations. Diverse community is perceived to be more productive. Diverse community is perceived to be more resilient to environmental shocks. So it is good for us to consider diversity from that perspective. It's very, very important to always measure diversity, because if a community is diverse, it means that it is healthy. What are some of the advantages? If a community is diverse, what is its advantage? Diverse habitat provides forage for a variety of invertebrates and vertebrate species. System stability results from species in the plant community being able to survive drought, insect plagues, and or disease outbreaks. And as a result, such a system ensures soil protection. It gives the system a lot of food in terms of forage. Such a system is more resilient to environmental perturbations. Genetic diversity also represents the potential for species to adapt to changing environmental conditions, which is considered as an important aspect of community stability and resilience. When a system is diverse in terms of at genetic level, such a system is more stable. Such a system is more resilient to environmental shocks, to any environmental perturbations, such as climate change, etc. Diverse community also has these benefits. When community is diverse, it means that such a community has a mixture of plants. And when there's different plants, soil is improved because some of the plants may be able to fix nitrogen. Those that are deep-rooted are able to translocate nutrients from soil layers below other plants' roots. Nutrients that are leached beyond the reach of shallow-rooted plants are able to recycle. They are able to come to the air horizons for shallow-rooted plants to utilize them. So it helps in translocation of nutrients. This is an advantage to such a diverse community. It also helps interactions. Interaction between some species are mutually beneficial, or at least not detrimental. We have interactions such as associations such as commensalism, symbiotic association. When there's such association, it gives the community a form of resilience, stability. Plants that are able to fix nitrogen. Some plants, let's take an example, symbiotic associations. Rhizobium species fix nitrogen directly from the atmosphere into the soil, while the plant's nodules also accommodate the organisms. This kind of association is symbiotic in nature. This is symbiosis association. From theoretical point of view, diverse plant communities generally have all natures filled. Because of that, such a community is less likely to be invaded by noxious or opportunistic introduced species. What I am trying to say is that if a system or a community is diverse, there's no room for introduced species. There's no room for invasive species. And that is an advantage. Diversity can be measured or monitored at several spatial scales. We can measure diversity at the level at alpha scale, the alpha level, beta level, and gamma level. So we have alpha diversity, we have beta diversity, and then we have gamma diversity. These are often used to describe differences in diversity across different spatial scales. When we are looking at diversity or measuring diversity at alpha level, what does it mean? It means that the alpha diversity refers to richness and evenness of species within plots or within sites. Alpha diversity reflects both the number of species and their relative abundance, and this can be considered within habitat or within plots. let us take a critical look or conceptual representation of alpha diversity. When you look at this, we see that this could be a quadrant or a very small area or an entire ecosystem. This could be, let's assume this is an entire ecosystem or landscape. When you lay plots within one location or within a habitat, this small plot, when you assess diversity within this plot, we say that you are looking at it at alpha level. This is an entire landscape. You see different landscape where there is vegetation, where there are mountains. When you lay plot within this small plot, this habitat, and assess diversity, assess richness and evenness within this small area, we say that you are looking at the diversity at alpha level. Let us look at beta diversity. Beta diversity is between sites or between habitats. So when we are looking at the beta diversity, here we are comparing usually diversity between two different sites, site A and site B. We can also represent beta diversity to be diversity across different environmental gradients. Different environmental gradients. When you look at it, we can lay plot along this gradient, along slopes, and then look at how species varies along these gradients. So here we are looking at it. We can look at it between plots or between different sites. Or we can also lay plots along different environmental gradients and assess diversity along different environmental gradients. The environmental gradient, what we are talking about, environmental gradient, what we mean is that it could be along elevations, along the slopes. It could be maybe differences in moisture conditions. It could be differences in light intensity or any type of environmental factor. You could see that some species may have different diversities. Some areas may have different diversity across slopes, across different moisture regimes. Let us look at this. Along gradients, this is the abundance of species. This is the environmental gradient. When you lay different plots, this is plot 1, plot 2, plot 3, plot 4. When you compare diversity of species along different gradients, you will see that there could be shared species. Different plots may have different species in common. Some plots may have unique species, so that they find the species turnover. Here, when you look at this plot and this plot, you could see that there are no shared species between the first plot and the fourth plot. They are different. So when you do that comparison along different environmental gradients, along elevations, along slopes, we say that you are looking at it at what? Beta diversity. You are looking at beta diversity. So, in summary, beta diversity is diversity between sites or between different habitats. We can also consider beta diversity as diversity differences in species diversity along environmental gradients, along elevations, along different moisture regimes. Now let us look at the next level that diversity can be assessed. We have gamma diversity. What do you mean by gamma diversity? We are using the same diagram. Gamma diversity, we are comparing the diversity of multiple or different sites, multiple different sites in different locations. We are looking at the entire landscape. We lay plot here, assess the diversity. We lay another plot here, another plot here, another plot here, almost all multiple plots. We are looking at different sites. That is gamma diversity. So if you have patches of habitat and you want to see how species changes spatially, that is gamma diversity. So when you check the conceptual representation of gamma diversity, you could see that we are not considering just one habitat. We are not looking at diversity within just one habitat or between one habitat and another habitat. We are looking at multiple sites, different sites at landscape level. Now, let us look at what do we mean by diversity indices. What is diversity? Sometimes it is convenient to express diversity using a single number. Diversity indices makes it easy to describe the diversity of site without listing species. And it enables us to compare sites that occur in different biomes and do not have any species in common. The simplest metric of diversity is species richness. Species richness, as I've already defined, is the number of species per unit area. Species richness is simply the number of species on a site, and is often defined or is represented by the letter X. While counting species is intuitive, richness alone does not clearly reflect diversity, because diversity is also influenced by evenness or the relative abundance of species. So when we are talking about species diversity, is not richness. Species diversity considers both richness and the species relative abundance, or it considers both richness and evenness. Now, let us look at this diagram. When you consider this diagram or the graphical representation of these two plant communities, site A and B, site A and B. When you look at this diagram, you could see that both communities have 50 individuals distributed among 5 different species. We have 1, 2, 3, 4, 5. The two sites have the same number of species, 5 species each. It has the same number of individuals, 50 each. But you could see that site 2 is dominated by one species, that is the green cross. When you check site 2, if you check site 1, there is 50 individuals each. There's 10 individuals each for the 5 different species. But when you check site 2, the Green Cross had 42 individuals. 42 individuals. The diversity indices that we use, that we are going to talk about, that is the Shannon-Winnes Diversity Index and the Simpson Diversity Index, always tries to incorporate both species richness and evenness to express diversity. What is Shannon-Winnes Index? The Shannon-Winnes Diversity Index is the most commonly used index of diversity in ecological studies. The values ranges from 0 to 5, but usually it ranges from 1.5 to 3.5. What is the advantages of using the Shannon entropy or the Shannon index? The Shannon index is relatively easy to calculate. What that means is that it is not computationally demanding. It is easy to compute. 2, it is fairly sensitive to actual site differences. It is very sensitive to site differences. However, it has some disadvantages. The disadvantage is that it assumes that all of the species in the community are represented, which is not always so. The second disadvantage is that it is possible to produce similar values of age for sites that do not have similar diversity. What that means is that at times, in reality, 2 different sites may have different diversities, but it is likely that when you use the Shannon entropy, it may give the 2 sites similar diversity index, and this is a disadvantage. How do we calculate Shannon-Winnes index? In fact, this is the calculation. If you look at the formula, don't be overwhelmed by the formula. It's a bit complex, but it is not difficult. Don't be overwhelmed by the formula. The formula is the age is equal to the proportion, the age is equal to the negative sum of the negatives. This age is equal to the negative sum of proportion of individuals in each species multiplied by the natural log of the individuals in each species. So this is the summation of the negative sum of the proportion of the species times the natural log of the proportion of that species. This equation is the same as this. It means that the proportion of the individual in the community divided by the total sum of individual times the natural log of the proportion of the individual. So again, it's a little bit overwhelming. The formula is a little bit overwhelming, but please, let's see how the computation is done. Let's compare these 2 communities, community A and community B. In fact, these quadrats or these plots or these habitats have bears. These are bears, different species of bears. Community B has also different species of bears. It could be trees. it could be macroorganisms, it could be plants, it could be trees or birds, it could be wildlife animals, anything. When you check this plot, that's this community A, how many species, how many individuals? We have 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. There are 10 individuals, and that is the abundance. So the abundance is what? 10. How many species? All these birds belong to one species. These birds, 1, 2, 3, 4, 5, 6, 7. They belong to one species. So 1, this is also another species. 2, this is also another species. 3, and this is also another species. 4. So the number of species or species richness is equals to what? 4. What is the diversity? We will look at that. Then let's come to community B. The community B, how many individuals, or what is the abundance? 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. So the community B and community A has the same number of abundance, 10. What is the number of species? This and that belong to one species. 1, 2, 3. The whole of this belongs to one species, so 4. So the number of species in community B is 4. So it means that community A and B have the same number of abundance. They also have the same number of species. Let's see how we can compute the diversity of species between community A and B. The diversity of species in community A and diversity of species in community A and B. Let's look at how we will do that. Now, as I indicated early on, that don't be overwhelmed by the formula. It is very easy to compute. Let's look at the table. Let's look at the table. This is the number of birds. We have blackbird, we have kiwi, we have secretary bird, and then we have chickadee. How you pronounce it. So it means that blackbird, how many blackbird do we have? 7 in community A. We have 1 kiwi, 1 secretary bird, 1 chickadee bird. So what is the abundance? You just have to add this. 7 plus 1 plus 1 plus 1 is what? 10. So the abundance is 10. Now, what is the proportion of blackbird in the community? You just have to divide 7 by 10. If you divide 7 by 10, you will get 0.7. What is the proportion of kiwi in the community or in the population? It's 1 divided by 10, you will get 0.1. The same thing applies to secretary bird. 0.1, 1 divided by 10, 0.1. Now, we are done with this. The PI, that's the proportion of individuals in the community. We've computed for that. That is the column this column. Now, the next column that we are going to compute is the natural log of the proportion of the individuals. So when you multiply 0.7, when you take the natural log of 0.7, you are going to get minus 0.35667. You can take your calculator and try that. You take the natural log of 0.7, it's minus 0.35667. You take the natural log of 0.1, you will get minus 2.30259. So you repeat the same thing for these proportions, you get this. How do we compute for Shannon-Willis diversity index? How do we compute for the diversity? You have to add this. Take the summation of all this. You add all of these, you will get minus 0.94045. Because we have a negative sign here, this negative will cancel this negative. So in the end, we are going to get 0.94045 as our diversity index, as our Shannon-Willis diversity index. Let's look at community B. The community B with blackbird species is what? 2. We have 2 of them. Kiwi bird, how many? 3. Secretary bird, how many? 3. Chickadee bird, how many? 2. So when you add all these, you are going to get 10. Now, let us compute PI. In other words, let us compute the proportion of each bird within the quadrants. So 0.2, 2 divided by 10 is 0.2. 3 divided by 10 is 0.3. This is 0.3. And this is 0.2. You take the natural log of this proportion. The natural log, ln 0.24 will give you minus 1.60944. The natural log of this 0.3 will also give you minus 1.20397. You take the natural log of this, will also give you this. The natural log of 0.2 will also give you this. To compute for the diversity index, you have to add all this. You will get minus 1.36616. Now, when you look at community A and B, what do you see? When you look at this, it shows that even though the abundance of species and the species richness are the same for both two communities, that is community A and B, but the Shannon Diversity Index shows that community B is more diverse than community A, because community A gave us 0.94045 diversity. The diversity index for community B is 1.36616. So what is this telling us? What does it matter? It means that richness and abundance between communities may be similar. Two communities may have similar richness and similar abundance, or two communities may have the same abundance and richness, but different diversity. But the diversity may differ. That is why I said that Shannon-Winnet Diversity Index incorporates both species richness and evenness or relative abundance to determine diversity. Another index that I want us to look at is Simpson's Index. Simpson's Index. There are several different ways of calculating Simpson's Diversity Index. Simpson's Index measures the probability that two individuals taken at random from a population or from a data set of interest will be the same. That is the D. When you are dealing with the D measures the probability that two individuals taken at random from the data set or from population of interest will be the same. Now, when you look at the formula, I personally prefer using Simpson Index 1 minus D because it gives you an answer between 0 and 1, which is a lot easier to make sense of the analysis. The 1 minus D measures the probability that that is this one, the 1 minus D, this is the D. This is the D. When you are using this one, the interpretation is different. When you are using the 1 minus D, the interpretation is different. But I personally prefer this to this because it makes explanation easier. The 1 minus D measures the probability that any two randomly selected species from the sample or population will be different. The N, when you check this formula, the N is the total number of individuals or organisms. And the small n is the number of individuals of a particular species. The pi, which I have already explained, represent the proportions of individuals in each species. In fact, don't be confused about the formula. Forget about how complex it is. The computation is very simple. The 1 minus D measures the probability that any two randomly selected species from the sample or population will be different. 0, when you get the answer to be 0 or closer to 0, it means that the diversity is low. When the answer approaches 1, it means that the diversity is high. The D measures the probability that any two randomly selected species from the sample or population will be the same. Here, when you are using D, it means that when the answer is equal to 0, or when the answer gets closer to 0, it means that the diversity is high. When the answer gets closer to 1, it means that the diversity is low. Please don't be confused about the 1 minus D and the D. Don't be confused about that. So because of this, whenever you are doing any diversity measurement using Simpson Diversity Index, you should let your readers know whether you are using the D or you are using 1 minus D. It is very, 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 very important. Otherwise, you will confuse your readers. You will confuse the audience. How do we compute diversity using Simpson's index and determine the diversity for community A and B? Now we are going to use the same data set that we used for the Shannon-Winnet Diversity Index. Don't forget that Simpson's index measures species dominance. It measures species dominance. Now, let's consider community A and community B. Community A and community B. For community A, we are using the same data set. How do we compute for Shannon-Simpson index using this data set? The data were derived from this plot. 5 different birds. Blackbirds were 7 in number for community A. Kiwi 1. Secretary bird 1. Chickadee bird 1. So when you add the abundance, you get 10. I have already explained that. Now, we are using this formula. You can try the others. It's the same thing. This formula is what? D is equal to 1 minus the summation of the proportion of individuals, the square of the proportion of individuals of species within the population. So now, let us compete for the PI. To compete for PI, as I've already illustrated in the previous lectures, the previous illustration, 7 divided by 10 will give you this. You know how to do this computation. How we arrive by this, you can do it. That is the proportion of each bird within the population. Now, how do we compete for this? You square this. When you square this, you are going to get this. Check the formula. When you square the proportion of each species, you will get this. When you square 0.7, you get 0.49. When you square 0.1, you get this. When you finish, you take the summation of this. You get, when you take the summation of this, add all of this, you will get 0.52. And that is D. And this is the probability that any 2 individuals sample from the population will be the same. There's 52% chance that any 2 individuals sample from the population will be the same. So in other words, the larger the figure, the lower the diversity. But I don't want to use this. If you want to use the D, the 1 minus D, you have to subtract this value from 1. When you subtract this from 1, you will get 0.48. This means that there is 48% chance that there's a 48% chance of randomly picking 2 that are different. It means that there's 48% chance that any 2 species that will be randomly picked from this plot will be different. Now, let us look at community B. So community 1, what is the Simpson's index? The Simpson's index for community 1 is 0.48. Let us look at the Simpson's index for community B. The same procedure. Here we have blackbird, we have 2. Kiwi, we have 3. Secretary bird, we have 3. Chickadee, we have 2. So the same thing, the number of individuals in community A is the same as community B. When you add all this, you get 10. That is the abundance. The total abundance of species within this plot. Let us take the proportions of each species within the community. You divide this by 10, you get this. And you know how we arrived by these proportions. You divide each individual, the abundance of each individual within the plot by the total individuals within the plot. You get these proportions. To compete for the PI square, it means you take the square of this, the square of this, this and that, you get these values. You take the summation of these squares, that is the summation of these squares. When you take the summation, you are going to get this 0.26. This is not a formula we are using. This formula is the Shannon Diversity Index formula. This is the formula we are using, this. Okay. So here, the diversity is what? The D is 0.26. And this means that there's 22% chance that any 2 individuals that you will pick here will be the same. So it means that if you want to explain it in the reverse, you have to divide, subtract this 0.26 from 1. When you subtract 0.26 from 1, what will you get? You will get 0.74. What does it mean? It means that there is 74% chance that if you pick any 2 species at random from this plot or from this population, there's 74% chance that they will be different. So in other words, the diversity of species in community B is larger than community A. Because community A, there's 48% chance of picking 2 different species at random, whilst in community B, there's 74% chance of picking 2 species at random. It will be different. There's 74% chance that any 2 species that will be picked at random from this plot will be different. So in all, I hope this video is being useful. Thank you very much for watching.